Western blotting technique. Animation by Mangesh More, ABCP Sangli. Introduction. Western blotting is also called as immunoblotting technique. This technique is widely used for the detection of specific proteins from a complex mixture obtained from cells or tissue extracts. Detection of specific antigens or antibodies is possible with Western blot technique. Thus, it is used as a strong diagnostic tool in clinical medicine. It is used for detection of HIV infection. Steps involved in Western blotting. First step is separation of proteins from protein mixture according to their molecular weight and size by electrophoresis. Second step is efficient transfer of separated proteins onto solid support. This is called as blotting. Third step is detection of target proteins by appropriately matched antibodies. Procedure of Western blot technique. First, tissue preparation. Whole tissues or cells from cell culture are taken. Mechanical disruption is carried out. Mechanical breakdown of cells involve use of blenders or homogenizers. Sonicators are also used for the disruption of the cells. Appropriate salts, buffers, and detergents are added to facilitate the breakdown of cells. Proteases and phosphatases are inhibited by the addition of specific inhibitor substances. This makes sure that soluble proteins of our interest are not getting digested from the enzymes released due to the cytolysis. Tissue preparation is carefully done at cold temperature to avoid the denaturation and degradation of soluble proteins. A combination of biochemical and mechanical techniques comprising filtration, centrifugation are used to get the final tissue extract. Mixture of proteins obtained by this method is further subjected to denaturation. Proteins lose their tertiary and secondary structure after the denaturation is complete. Denaturation occurs as the disulfide bonds between the peptides are broken by the action of strong reducing agents. Denaturation is done by treating the proteins with heat, acid, or alkali. Strong acids and strong alkali break down disulfide bond. Second step is gel electrophoresis. SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis is most commonly used technique for gel separation. Protein mixture is separated according to their molecular weight and size under the influence of electric current. Steps involved in the gel separation. Protein mixture is added to the separation buffer. Separation buffer contains SDS, which induce negative charges to the protein molecules. SDS is the detergent. It covers or coats the surface of linear proteins, causing them to acquire negative charges. So all proteins acquire negative charge. Loading of sample on gel matrix. Protein mixture is added onto the cups or vials made at one end of the polyacrylamide gel. Other vials are loaded with equal amount of molecular weight markers and positive negative control samples. This is how the loading is done. First vial is loaded with molecular weight marker. Subsequent vials are loaded with test samples and control samples respectively. Gel chamber is then filled with buffer. The electrodes are then connected to battery unit and the power supply is turned on. The gel chamber is first filled with the buffer. Then the connectors are connected. This is how the unit looks like. A die front at the top. All proteins negatively charged move down in gel from cathode to anode. As they are negatively charged, they travel towards positively charged electrodes, so they trickle down through the solid gel. The electrophoresis is done till the diaphragm reaches the other end of the gel. When the diaphragm reaches the bottom, electrical supply is turned off. Electrophoresis is stopped. Third critical step is blotting or transfer onto solid membrane. After the protein separation is over, the gel matrix is transferred onto a membrane for electrophoretic transfer. The membrane is made up of nitrocellulose or PVDF, polyvinylidene difluoride. Gel sandwich is prepared as shown in the diagram. The gel should be facing towards negatively charged electrode, while the membrane should be facing towards positively charged electrode. The proteins transfer from the gel towards the membrane. Transfer buffer contains methanol, which separates SDS from the proteins, resulting into neutral charges to the proteins, which adhere onto the membrane firmly. After that, membrane is washed with washing buffers and subjected for further steps of Western blotting. Area of the membrane not covered by the protein sample is coated with BSA, bovine serum albumin or toned milk. This is called as blocking. Blocking blocks the unwanted antibodies attachment to the empty spaces on the membrane. This is the composition of blocking buffer solution. The blocking solution is poured onto the membrane. Last important step is detection. Antibodies specific to the protein of interest are added as a solution over the membrane, followed by incubation and washing. Primary antibody solution is added on the membrane. The primary antibody solution, after addition, incubation is carried out at room temperature for about 1 hour. After that, the secondary, that is the labeled antibody, is added, followed by the incubation. The secondary antibody solution is added and the incubation is carried out. Secondary antibody, which is the labeled antibody, helps in the detection. Detection, it might be colorimetric detection. If it is a colorimetric detection, the secondary antibody is linked with the indicator enzyme like horseradish peroxidase. Use of chromogenic substrate will directly help detection of protein of interest, as chromogenic substrate will produce colored end product. Chemiluminescent detection, if a secondary antibodies are linked with the enzyme that convert the substrate to a luminescent product, chemiluminescent end products are the sign of presence of protein of interest. There are other techniques also: radioactive detection, fluorescent detection. In case of radioactive detection, a radio labeled secondary antibody is used. The radioisotope will cause formation of image on the X-ray film directly. So X-ray film is placed near to the Western blot membrane and the image is produced. In case of fluorescent detection, exciter wavelength will generate fluorescence. The presence of fluorescence is detected and considered as the sign of presence of protein of our interest. This is it. Hope you like this video. Thanks for watching.